Now, we would like to introduce you to John Ferbas and Xianson Xie from Amgen. John has been with Amgen for more than 20 years and currently works in the Biomics Group as the Director of Cytometry. Dr. Xianson Xie holds more than 20 years of experience in cell biology, microscopy and imaging. He is a senior scientist at Amgen, where he primarily serves to leverage microscopic and imaging applications and approaches to support various projects in both therapeutic discovery and in the clinical space.
Hi, everybody. My name's John Ferbas. I'm the director of the Cytometry Sciences Laboratories here at Amgen. I'm here along with Jinsong Xie to describe our experience using the NanoLive platform for lipid droplet analysis. This fits well into my accountabilities at Amgen, where we use a variety of flow cytometry analyzers, sorters, single and multiphoton microscopes, bioluminescence platforms, and now most recently, the label-free platform provided by NanoLive. Before going on, it's important that I mention to you that no one at Amgen has received any compensation for NanoLive from this work. Likewise, Amgen discourages us from endorsing specific platforms, reagents, or consumables, but rather encourages us to emphasize the science that such approaches can release. Stated another way, there are many ways to answer scientific questions. The NanoLive platform was our choice to interrogate lipid droplets, and that's what we're going to share with you today. Now, back to the science. We all know that our bodies are comprised of trillions of cells that grow, die, and interact as an ecosystem or a community. I've spent my entire career studying such cells in the setting of the life sciences and the pathologies that can be driven by them. In the beginning of my career, I spent essentially 100% of my time using fluorescence probe-based methods and flow cytometry to accomplish these goals. Now I'm about 50-50 with respect to flow cytometry versus microscopy. Most recently, label-free approaches have really excited me, all the way to the extent that this has really become part of my 5-year strategic plan for the laboratory. This is because probe use is not benign. And with the topic of lipids in particular, we know that the introduction of probes can create artifacts not only with the dynamics of lipids within a living cell, but they can also induce phototoxicity if one is interested in obtaining time-lapsed studies. So when we initially were talking to scientists in the core that had the need for us to mount lipid droplet analysis campaigns, of course, the first thing that I considered was fluorescence microscopy. But as I said, I was very wary of this approach because I knew that probes and lipids do not match, do not mix, aren't compatible. And I also knew that if I wanted to do time-lapsed images, that I would be holding the liability of phototoxicity as a function of the probe. I also have a great interest in Raman scattering, and of course that immediately came to mind for investigations such as this. But I don't currently have the infrastructure built to do Raman scattering, and I knew that I would have to hire dedicated staff to accomplish these goals. Stated another way, I knew that if I committed myself to Raman scattering, I'd never really be able to enable an investigator to come into a lab, take their measurements and their data, and go back to their desk. So that took me to the last row on this table, which is this paper that was published by Sandoz et al. That brought forward the strengths of the NanoLive platform. It doesn't use probes. It's immune to phototoxicity. And even in the postage size video that's in the bottom right corner of this slide, you can see that lipid droplets are clearly distinguished by a function of their refractive index. And you can even appreciate the dynamics in this very short video. So As the director, the manager, and the administrator of the lab, it's time for me to hand the podium over to Jinsong Zi. He's the scientist in the lab that did the experiments, that had the direct experience with this, and the most appropriate person to relate what he has done with you. Thank you.
Hello, everyone. My name is Jinsong Zi. I'm a senior scientist working at Amgen Cytometry Sciences Group. I have over 20 years of experience in microscopy and imaging. At Amgen, my primary role is to leverage microscopy and imaging platforms to support various projects in both therapeutic discovery and clinical space. As a follow-up of the big picture provided by Dr. Ferbas, next, I'd like to speak to the at-the-bench experience with smart lipid droplet assay, where I was an alpha tester. Since these early efforts, I'm pleased to say that Nanolive developed this to a point of one-click assay. No labeling, no centrifugation, just some high-quality cell culture, minus and plus treatment of interest, followed by data collection. Now, when it comes to lipid droplet, their characteristic refractive index allows the very clear and accurate segmentation of these lipid droplet particles from the remainder of the cells. As shown in this slide, which I reviewed as the perfect first application for us. I was very excited to try it, as we have many investigators with a need to study lipid droplet in a variety of cells. So now, with a little experience under my belt, I often go home after loading my experiment on this label-free platform. And when I come back to work the following day, I can just use this AI-anchored Eve analytics software to extract up to 40 different useful metrics. So today, I will not have time to review all of these metrics with you. Rather, I will refer you to the very helpful technical notes provided by Nanolive for detailed information for you to better understand the capability of this assay. So before moving forward with Nanolive data, I wanted to remind you that if I use a fluorescent probe and perform a time-lapse imaging experiment on a standard microscope for a few hours, I will see photo bleaching and photo damage. I don't need to worry about any of these problems using this label-free platform. As shown in this slide, in this case study, time-lapse images were collected on a Nanolive CSA system every 90 seconds for a total of 4 hours on a pancreatic cancer cell line called Danji cells, with and without OA or oleic acid treatment. As shown in this video on the left, oleic acid treatment can clearly cause the accumulation of lipid droplet in these cells. In my career, with over 20 years of microscopy, I have seen many segmentation tools, and I can tell you that this segmentation analysis provided by Nanolive is the best segmentation I have ever seen. Another thing this assay really impressed me is these parameters of dry mass reported from this platform. As you know, dry mass is everything other than water content in the cells. So we know that if we load the cells with oleic acid, we're going to see increased lipid droplet content and also increased dry mass. As shown in these time normalized graphs on the right, we can clearly see that oleic acid treatment can cause an increase in the mean dry mass content from these cells. This is also detected in the population scale in the total dry mass of the cell and in the total lipid droplet dry mass. So without this label-free platform, I will never be able to collect such meaningful data using a standard microscope. I do want to point out that in this experiment, oleic acid treatment was used as a proof of concept. So I will leave it up to your imagination to think about what if you use your therapeutics of interest to treat your cells and then use this label-free technology to look at your cells and see how your therapeutics will affect lipid droplet dynamics in your cells. Another attribute I found very helpful from this assay is that in addition to obtaining very helpful time-lapse imaging result, the assay also enable me to acquire very comprehensive static images from the assay. And I really like the flexibility of the assay output. To summarize my experience with the use case scenario, I would like to highlight 4 very important take-home messages. First, the instrument has a very small footprint. It allows us to put it anywhere in the lab. Second, I don't need to spend a whole day to label and process my cells. Third, I can easily train the assay to someone and allow them to do their experiment independently. And lastly, I can simply just load the experiment before closing my day, and I can just get very meaningful data after coming back to work the following morning. Lastly, I wanted to thank my Amgen colleagues who has been very instrumental and help enable me to do this work. I also appreciate the help of Sanila and Matthew from NanoLive for their support and being the key persons making this alpha test possible. Thank you.